Um, so as Christian mentioned, I'm going to be focusing more on structural analysis, and this will be at the protein level, not the DNA or RNA level. And I hope that I can convince you that this is an example of a case in point for moving beyond, as the title of the mini symposium is, moving beyond sequence alignment to try to get an understanding of an important phenomenon in biology, which in this case is proteome oxidation in bacteria. So first of all, I want to, for those of you who are not familiar with this phenomenon, I want to give you a motivating slide on why we care about proteome oxidation in the first place. Starting with my model organism, Deinococcus radiodurans, which is the most, of known species, it is the most highly radiation-resistant organism. Ionizing radiation produces reactive oxygen species, which in turn causes proteome damage. And something is known in terms of the importance of proteome oxidation in determining the resistance of this organism in general, but molecular properties which underlie this resistance have yet to fully be elucidated. I'll be hoping to do some of that here, at least make a first stab at it. This phenomenon is also important for understanding the process of desiccation in all cellular life. Here I've just shown an example of a plant before and after a massive drying event. And this is because reactive oxygen species concentrations locally increase as you lose solvent through desiccation. Proteome oxidation also has important implications for understanding a number of age-related diseases in humans. I won't go through this entire figure here from this recent review, but just to highlight a couple, this includes cancer, certain specific types of neurodegenerative disorders, and metabolic diseases. And then finally, for the synthetic biology community, this has important implications in understanding unintended side effects, usually in the form of metabolic shifts that can result producing ROS more so than in a wild type state, and in the massive, perhaps even overuse of fluorescent proteins in various experiments, which themselves lead to generation of reactive oxygen species through chromophore maturation, the mechanism of which was recently worked out. Yes?
Is that I know people are watching in single molecules, watching proteins being produced and moving in the cell. Is this, the fluorescence, affect the biological activity when connected to the imaging?
Well, what I'm referring to is less connected to the imaging itself and more so a side effect of simply using fluorescent proteins for imaging.
Yeah.
Right, so with high concentrations of these molecules, you of course get high concentrations of ROS that are part of the maturation process of the chromophore, which can lead to deleterious effects on the cell, if you're working with live cells, for instance.
But the short-term effects connected to the imaging is
not so much of concern. Sure. So a little bit of the biochemistry involved here. The specific reactive oxygen species that I'm concerned with are the superoxide radical, hydrogen peroxide, and hydroxyl radical, which can lead to these specific modifications on the side chain of lysine, arginine, proline, and threonine residues. And that is the formation of this carbonyl group on these side chains. These are in turn highly reactive, as are reactive oxygen species, and can lead to a variety of downstream modifications in a protein. But in general, what is observed is a massive misfolding of proteins that happens progressively with prolonged exposure to ROS, proteolysis, and protein aggregation, which, if extensive enough, can lead to cell toxicity. So I'm going to try to convince you before going further that the phenomenon I talked about before of radiation resistance in Deinococcus radiodurans is largely tied to its ability to avoid this type of proteome damage. compared to sensitive species. And so what I'm showing here is a plot of survival rate, which is represented by this curve with dots or circles here, in blue for radiodurans, in red for E. coli, and then overlaid on this is in triangles the accumulation of carbonyls in the proteome for both organisms. And what you observe here is an inverse relationship between accumulation of carbonyls and survival rate. Interestingly enough, the level of accumulation of these carbonyls at the crossing over point between survival rate and accumulation is actually quite similar between Deinococcus and E. coli, which is further substantiated here when you look at the killing rate as a function of this accumulation. And when you overlay data measured for E. coli in red and again blue for Deinococcus radiodurans, you see almost the same trend exactly. And also, this is irrespective of the type of ionizing radiation. They show both UV exposure and gamma exposure here. So this is fairly strong evidence to suggest that carbonyl accumulation in proteomes is actually directly tied to the killing and or survival rate, as it may be, of bacterial cells. So it is of interest then to identify specific targets, both in terms of proteins and even specific residues on proteins that are likely to get hit by ROS in cells. And there exists a methodology called Redox Proteomics, which is a mass spec approach to identifying residues that get modified by ROS and form carbonyls. But there are some problems with this approach. First of all, the inherent transience of these carbonyl groups, which are caused because the secondary reactions that I alluded to before, lead to a loss of the carbonyls and they transform into something else and cannot be detected, as well as the proteolysis and aggregation events, which obscure these events from experimental detection. So that's a bit of a problem. Secondly, the experimental platform itself can lead to some loss of measurement capability as well. On the one hand, because a chemical label, which is often used for stabilizing these inherently unstable carbonyl groups, also interferes with tryptic sites, which are required in order to perform proteomic mass spec. And there are enrichment strategies as well to try to more specifically bias measurements towards oxidized peptides. But what you end up doing is ruling out things that either aren't oxidized or are oxidized to a much lesser extent, and you miss those events. On the data side, the net result of these factors is that you end up with a relatively small number of positive residues that form carbonyl groups and a relatively higher number of lower confidence negatives. So it's desirable to get some theoretical understanding of the underpinnings of this phenomenon. And there have been a couple of examples in the literature using a sequence-based analysis approach to try to predict sites that are more vulnerable to this type of damage. I won't spend too much time on this, but I just want to give you a sort of flavor of this before I dive into mine for comparison. So there's this method, CSPD, that has not complete residue resolution predictions, but predicts a region that might be sensitive. And the basis for this is a sliding window where they look for sequence motifs, enrichment of the sensitive amino acid types, inclusion of at least a proline, which is the most reactive of these 4 amino acids with ROS. And a couple of things that start to get at molecular properties like iron binding sites, which is important because of the possibility of metal catalyzed oxidation, and then hydrophobicity and solvent accessibility. So they're starting to get at the molecular properties, or attempting to, but it's still very much rooted in sequence space. They had a relatively small training set here, and what you end up, so I took their own training set and ran their own predictor on it and generated this confusion matrix here, and what you see is that the true positives they predict are actually very, very limited in terms of the actual positives that were measured experimentally. And they end up settling on a model largely because they get a lot of true negatives, which we don't really care about so much when the question is what is rendering protein molecules to be vulnerable. So we care more about this value here. So they largely miss the point here. Another later predictor, again rooted in sequence space, at least gives us single residue resolution and is again searching for local motifs in sequence space. And it uses two metrics, position-specific propensity of amino acid pairs, as well as Kamer diversity, that have a relationship to local homology. And this AA metrics is another attempt to kind of move towards molecular property space. But they end up, again, so in this case this was applied to mammalian proteins, but again they end up very likely under-predicting the true positives here and rewarding themselves for getting a lot of uninteresting true negatives correct. And I say, after having reviewed these existing efforts, there are some important lessons to be learned in going forward and trying to do better, which include that these sequence-based methods are related to the local homology of a relatively small training set and would not be expected to generalize very well when trying to look at evolutionarily distant proteomes or even just specific proteins within the same proteome in terms of their vulnerability to oxidation. We also see that perhaps they have not done enough to try to address the data imbalance issue of few positives to many negatives. And then even if these predictors performed very well, they don't provide a lot of fundamental insight into the molecular properties underlying the phenomenon. And therefore, there's not too much we can do with output from these predictors in a prospective sense. If we want to, say, search for interventions or use it in engineering efforts to design resistant proteins. Okay, so my approach is motivated by the sort of intuitive observation that structure is what stratifies the vulnerability of reactive oxygen species across a proteome or between proteomes. And I just show in this cartoon here a little bit more about what I'm talking about. Taking these four distinct protein structures and identifying the RKP and T residues, which are the oxidizable residues, and thinking about the difference in the molecular properties of each of these molecules as well as the distribution of these sites within them, we can imagine that the integrity of the native fold of these structures differ across them in terms of increasing exposures to oxidizing reagent. The less exposed there are fewer RKPT residues, of course, we're going to have a higher propensity to preserve the native fold, and the more extensively they are composed of RKPs and Ts, especially if they're exposed on the exposed surface, we might expect that this would lead to rapid unfolding of the protein. So I'm interested in taking this sort of theoretical approach and looking at molecular properties that have both been highlighted in the literature and come from my own intuition to see if we can rigorously develop a predictor around said molecular properties to identify vulnerable sites within proteins and within proteomes. Okay, so I'm going to give a brief overview now of the overall workflow, and then we'll kind of touch on each of the components of it. I don't have enough time to go into full detail of each part. First of all, we start with the proteomes of interest, in this case Deinococcus radiodurans and E. coli for my study. And ideally, we want to have a structural representation that's rooted in experimental determination. And for E. coli, we get more than 50% coverage of the encoded proteins by crystal structures contained in the PDB. But for radiodurans, the coverage is far, far lower. So if we want to attempt this, we have to do high throughput structural modeling. And I'll describe that first. Next, formalizing these molecular structures as features mapped to 3D space that I can train a predictor on is sort of the next step. And the bioinformatic methods that are previously published that enable this step are actually largely rooted in sequence analysis. On the experimental arm of the workflow, we have this Redox Proteomics, which I alluded to earlier, that allows us to identify with site specificity or residue specificity where carbonyls form and where they don't form. And then performing single molecule analysis on these peptides allows us to derive a training set, which will then couple with these molecular features as input for a machine learning algorithm, which I'll then use to predict sites proteome-wide. And then finally, a holdout set from this training set will be used to validate the prediction and offer the beginnings of insight into molecular mechanism underlying the phenomenon of protein oxidation. So we'll start with the structural modeling first. I took a composite approach to modeling all of these structures in the proteome, using, of course, where available, PDB structures that were determined experimentally, but supplementing that with multiple different algorithms that have completely different bases, including ITASR, which is the top performer at the last couple of CASP experiments and is a local homology method. ProteMod server implements a couple of global homology methods. Quark and EV-fold constitute ab initio folding methods, sort of from first principles. The distinction with EV-fold is that it's constrained by co-evolving positions within proteins, and this is a method that was developed in my department by Debbie Marks's group. Give us an idea about what we can expect for the performance of these algorithms on the radiodurans proteome. We see here that the breakdown of the proteome in terms of the length of the proteins, as well as available high-quality templates that cover these, which enable homology modeling methods. And you can see here in green that more than 60% of the proteins encoded are seen as easy to model by homology, which is nice. Another 20% or so, 20 to 25%, fall into a category of having at least some high-quality templates for homology modeling. And then in red here, we see the minority of the group has no available structures to enable homology modeling and is only approachable by modeling using ab initio folding methods. So the good news is that the worst of cases, which lies here with no available homologous templates and very long structures, or very long proteins, is actually quite small. So this is where we need to be worried about what we'll miss, but at least it's a very small fraction of the proteome. Okay, so the next question is, well, I've run all these methods and I have a number of alternative models for every single protein. How do we discriminate between them, and when do we say we have a good enough model to feed it into downstream analysis? So I have a number of criteria that I use to discriminate between them, including the coverage of the target protein, whether or not it has a computable surface using the software package UCSF Chimera, which I use for a lot of my feature computation, stability scores. These are sort of standards in the feature for assessing quality of structures. TM score, which is like a proxy for how similar to homologous crystallized structures a given model is. The percentage of residues that are in favored positions compared to a Ramachandran plot, and then a G factor, which is like an overall conformation score for a protein structure. And what you see here is a summary, a broad summary of the models that I generated for Deinococcus radiodurans and the fraction or percentage of them that satisfy these criteria here. And you see that the vast majority of them satisfy all of these criteria with the exception of TM score, favored residue positions, and G factor. And then the question is, well, how bad is this? Comparing to what exists for Deinococcus radiodurans in terms of actual crystal structures, you see a fairly similar distribution with respect to satisfying these criteria. The main exceptions, again, are these three factors down here: the overall conformation quality, favored positions for Ramachandran plots, which, by the way, I think is notable for PDB structures, isn't 100% here. And then TM scores, which is actually lower than for the models, and the reason for that is these 82 crystallized proteins for radiodurans are not homologous to other things in the PDB, so they can't resemble much that's also contained in there. This is a breakdown of the performance of these different approaches to actually deriving the best representative structure. About 50% of them are covered by ITASR. Another third or more than a third of them are covered by the global homology modeling methods. A cut down here satisfied by ab initio folding methods. You can see the small wedge of available D-radiodurans PDB structures. And then I alluded to before this difficult region of proteins that consist of longer proteins with no homologous templates that we end up with no representation of in the data. Next, I'll touch on molecular feature engineering that will feed into the machine learning pipeline. So from the literature and my own intuition, this is the collection of properties that I'm interested in investigating. I should note that this first group is only determinable based on having a 3D structure at all, which include the depth beneath the surface of the protein, solvent exposure, surface cysteines and methionines that can act as sort of local sponges for ROS in a reversible manner, contacts and circular variants, which is like a proxy for molecular density. Then we have properties that can be determined from having the amino acid sequence alone, including secondary structure, disordered regions, potentially protein binding regions, and also metal binding sites. And finally, we have properties that simply are known based on the identity of the amino acids in question. And I won't list all of these for now. And the next question is, how do we featurize these in a way that we can then feed into a machine learning approach? So basically what I do is I parameterize all of the molecular properties I just showed you on the previous slide using the following set of potential parameter settings. Grouping, this doesn't apply to all properties, but applies to those such as secondary structure, where you could say one property is this residue included in a secondary structure or not, versus this residue is part of an alpha helix or a beta sheet. So you can use a composite or separate approach for those properties. For data type, there's binary and continuous. If you have, say, a probability score for disorder or just a binary call for this residue is in a disordered region or not. Spatial scale, and this really requires having a 3D model or experimentally determined structure. So at the smallest scale, it's the atom that actually can bear the carbonyl if oxidized, and then you have intermediate local scales, and then the global scale of the entire protein to summarize these properties. And I use a summary statistic of either the sum or the mean, and I test both for every single molecular property at every single scale here where applicable. And what I end up with is taking all of the properties I listed before, almost 400 unique features. The next step is the experimental arm here. So briefly, the experimental approach begins with actually irradiating these bacterial cultures using a custom rack that I designed that withstands gamma irradiation while not blocking its transmission, and zapping them for a couple of hours to give a gamma dose of about 6.7 kilogray, which is more than 3 times the amount necessary to completely sterilize E. coli. So you get no surviving E. coli cells, and is enough to measure an impact on radiodurans with a survival rate between 50 to 70%. The next is extracting the proteins from the cells and derivatizing them using a chemical reagent, dinitrophenylhydrazine, which selectively reacts with the carbonyl sites here to yield these adducts. And this stabilizes this position, and because it's selective, we know that when we measure the resulting peptides by mass spec, that the mass shifts observed because of these adducts are actually due to oxidation at these sites. And here's just a brief overview of the data that we get out of this. Again, I mentioned before what the sort of technical limitations of the experimental approach are. So we end up with, instead of potentially what I would hypothesize would be maybe an order of magnitude higher than this, perhaps, we end up with about 100 proteins in E. coli and about 70 proteins in radiodurans that have at least one peptide that has been oxidized in them. And if we're looking at the residue level, we see this imbalance again between positive sites and negative sites that were detected on peptides that were oxidized at least one site. And I might note here that these sort of faded or lighter shades represent fractions of these data that only were measured after gamma irradiation and are going to be my holdout set for final validation of my model. So a quick coarse-grained overview of the functional relevance of protein oxidation just from the data I showed at the protein level was analyzed through gene ontology term enrichment analysis. And this uses a hypergeometric distribution to essentially look for enrichment of annotation terms within groups of proteins, in this case oxidized proteins. And what we see for E. coli is that there's significant enrichment for terms related to translation. A lot of these actually turn out to be ribosomal proteins, and then some that have to do with small molecule metabolism, in particular primary metabolic pathways. And for radiodurans, we see recapitulated the same observation of proteins involved in translation selectively getting oxidized. I'll come back to this a little bit later. Next, I proceed with single molecule analysis to get my training set for machine learning. But before I do that, I want to talk about some theoretical questions that can be addressed even without the machine learning. It was previously observed in a paper that focused largely on the molecule bovine serum albumin that there is spatial co-localization of carbonyl sites that are measured experimentally. And this is conserved as you increase the exposure to reactive oxygen species. And the authors hypothesized that this was because of progressive misfolding of structures. So you hit one site near the surface, maybe it unfolds and exposes an initially buried site, and so on. And looking at this a little bit more rigorously, I looked at the distribution of all pairs of experimentally measured carbonyl sites versus pairs of one carbonyl site to a non-carbonyl site, and then pairs of non-carbonyl sites. And you see that indeed the distribution of interatomic distances is smallest for the pairs of oxidized sites. So I was interested in seeing if there might be an alternative explanation beyond just the kinetic process of misfolding, of progressive misfolding. And so in my single molecule analysis, I looked at every individual peptide and identified the oxidized residues and the non-oxidized residues. I mapped those onto my 3D structures and computed the interatomic distances of all of these pairs of residues. And we see that, first of all, we see that proteome-wide, instead of just looking at one protein species, this observation is conserved, that oxidized sites do indeed co-localize in 3D space. But then can we learn something else from this? So before I go into the conclusions, I need to give a little bit of background on the data side. This is a kind of fundamental problem in correlation analysis, which is that correlations that are computed are inherently unstable with small sample size, and that's illustrated nicely in this paper here. And this is a problem because the set of pairs of carbonyl sites is relatively small and much smaller than these other two sets I want to compare against. So what I need to do is identify the range of sample sizes where we can say beyond that we believe in the comparative correlations that we find here when comparing interatomic distance correlated with individual or combined features across these sets. But then also, okay, so the approach to resolving this is basically a bootstrapping approach to identify the point at which correlation stabilizes as a function of size. So I performed this type of analysis on all of the roughly 400 features, looking at interatomic distance and how well that correlates with individual feature distance. And what I see here are examples of individual properties like the disorder of a residue, where the carbonyl-carbonyl site pairs show significantly higher correlation with distance in the disorder space distance when compared to these other two sets. And that suggests to me that this may be one property that might be informative in predicting vulnerability to oxidation. And as a counterexample, the contacts within a 12 angstrom radius show really no difference between the carbonyl-carbonyl site pairs and these other two sets. So I used a greedy algorithm to combine some of these 400 features into 400 standardized features here, and found that this combination of features shows an even stronger correlation and a stronger separation between the carbonyl-carbonyl site pairs and these other two sets, which suggests a potential molecular signature for vulnerability to protein oxidation. And then, just as sort of a sanity check, I performed a similar procedure on randomly generated features, which show that really you cannot randomly produce this kind of difference with random features. And so the main conclusion that I make from here, I'm an avid music listener, so I like to summarize this as a combination of song titles. Born Under a Bad Sign, or Everybody Hurts and Not, I Am a Rock. And what I mean by this, Born Under a Bad Sign represents that there is indeed a molecular signature that seems to underlie vulnerability to oxidation. Everybody Hurts, meaning that all other sites maybe are just randomly lesser so vulnerable. And there doesn't seem to be, at least within the features I'm looking at, there does not seem to be a counter signature for robustness. Okay, we'll talk briefly about machine learning. I'll kind of skip over this. This is just an overview of the positive and negative training data. I will just mention that I pulled in data from the literature for an additional species which is slightly radiation resistant, at least to UV radiation, Photobacterium angustum, to increase my data set size, although we still retain this imbalance between positives and negatives. So I'll go on to summarizing my overall machine learning approach, which starts with the standardized roughly 400 features, which I engineered. And I'm using a support vector machine algorithm with a linear kernel as implemented in the scikit-learn Python package to define a classifying hyperplane in this multi-dimensional space to say whether a residue is vulnerable versus not. I use a scoring metric called the area under the receiver operating curve, which is standard in data science with predictions. Although many previous studies have tried to develop predictors around accuracy, which is notoriously bad for imbalanced data sets. I use weighted scoring to further improve this choice of scoring metric to deal with this imbalanced data set problem, so that the total set of positives in my training set rewards the predictor equally to the total set of negatives. And then I use cross-validation, running 1,000 trials with 5-fold splits. And I use a stratified shuffle split strategy, the stratified being important here, meaning that for each fold in a 5-fold split, the same ratio of positives and negatives is preserved as in the entire training set. And then I proceed with a parameter optimization grid search, which utilizes this cross-validation strategy, and a recursive feature elimination step to get rid of redundancies and really pare down the set of contributing features to those that are essential. And here's a brief glimpse of the results of both the parameter grid search and then the recursive feature elimination. I just show here that the AUC is used on the y-axis here to select the best combination of parameters for the algorithm performance. And then here I used a greedy algorithm with recursive feature elimination to find what the optimal subset of features was to get the highest performing predictor. And this corresponds to 30 features. And here, I don't have time to talk about all of the features and how they contribute to the prediction, but I'll just briefly highlight some of the top 10. And these are ranked, so using SVM with a linear kernel allows you to get an idea of the relative contribution of features based on the coefficients, which are the magnitudes of the coefficients of the defining hyperplane. And so the highest contributor is local average hydrophobicity. Polarity is also quite important. Rate constants for reaction with hydroxyl radical, and these other features here. One thing that I want to point out is that most of these are quite distinct from each other in terms of actual molecular concepts or molecular properties, highlighting that this is likely to be a complex problem that any individual molecular property probably cannot adequately predict. And then we'll move on to the validation step. So first of all, taking the full training set, retraining and testing on it, we see that we get an AUC of 0.72, which is this blue curve here for the final predictor, as compared to the performance of each individual feature used as a predictor, many of which alone do perform better than random at least. And I'll highlight here that the two sequence-based approaches that largely do depend on local homology don't generalize well at all to my data set. They are no better than random, really. Then the question is, well, here I'm just testing on my full training set. What about the real test, which is testing against an independent test set that was never seen during machine learning? And so I reserved this gamma-only test set that I mentioned before from my experimental data, and then tested the performance of this same classifier on it. And although there is some loss of efficacy, I still get a reasonable predictive capacity, whereas, again, both of the sequence-based approaches are really no better than random. They don't generalize well. We can also look at this at increasingly higher molecular scales. So moving from site to protein scale, we see that if I compute a value of carbonyl site enrichment that I predicted versus those that were measured experimentally for proteins, we see a correlation, a fairly strong rank correlation, looking at the Spearman row here, for E. coli. And these dotted lines represent the 95% confidence interval here. Similar result, although not quite as statistically significant result for radiodurans, where most of the points here do lie within the 95% confidence interval, but the regression line is not quite as good of a fit as it was for E. coli. One thing I want to point out here is that you'll notice that just looking at the scale of the x-axis and y-axis, I'm predicting on average about a 10-fold higher enrichment for sensitive sites relative to what's measured experimentally. Although that's maybe not necessarily so far from the truth, given that we already know we're undersampling real extent of protein oxidation given experimental limitations. So now let's get to some potentially interesting biology concerning this. We're able to now compare within proteomes for an organism and even across proteomes from different organisms based on this theory. So looking at the enrichment of carbonyl sites in orthologs mapped between Deinococcus radiodurans on the x-axis and E. coli on the y-axis, we can see, first of all, some broad conclusions. One is that you don't actually see a significant global enrichment for carbonyl sites in E. coli versus radiodurans. So it's not that this is a global phenomenon of genomically encoded protective motifs for radiodurans over E. coli. And even within each of the proteomes, you see that functional classes that we know are important for surviving irradiation and oxidative stress, like the ability to regenerate the proteome through ribosomal proteins and other translation-related proteins, as well as proteases to degrade the damaged proteins, DNA repair because DNA also gets damaged under these conditions, and ROS detoxification enzymes are mostly not so much outliers from the overall population of proteins. The one really significant thing we see is the high enrichment of sensitive sites or vulnerable sites within ribosomal proteins, which recapitulates the observation that we made in the experimental data. And I want to highlight a couple of things here. I ranked the ratio of E. coli enrichment in carbonyl sites to carbonyl site enrichment in radiodurans, and at the top of the list, at least with respect to these functionally categorized proteins, we see a ribosomal protein in which there's a twofold enrichment for vulnerable sites in E. coli versus radiodurans, and that's this 50S ribosomal structural protein L32, which is a non-essential protein. And then another one near the top of the list, which with a similar advantage for radiodurans over E. coli, is another non-essential ribosomal protein L36. And I was interested in seeing if anything was known about these proteins. And I turned up a knockout mutant screen for E. coli from this paper just a few years ago in JBAC-T, where they identified these two exact proteins in knockouts as generating conditionally sensitive strains to ROS exposure. So that potentially constitutes some key proteins that could be involved in radioresistance, given that, if my prediction is correct, the radiodurans orthologs of these are especially, or rather not especially robust, but rather less vulnerable. So in summary, the things that I'd like to say that we have learned from this are, one, although I would not say that on a protein-to-protein basis we can have full faith in structure modeling methods, at least it is a tractable problem in that we can get near full coverage of proteins and seems to be useful in some contexts. I also presented a novel framework for taking molecular molecular properties and translating those into 3D space to enable a machine learning approach to looking at a post-translational type modification to proteins. I substantiated a new hypothesis to explain why carbonyl sites tend to co-localize in 3D space, which is irrespective of the kinetics of progressive misfolding. Simply that if they're closer together in 3D space, they very likely share a local molecular environment. And if that environment is sensitive, then multiple sites within that space may be hit by ROS. We also established some evidence that there is likely a very complex molecular signature that underlies this vulnerability, and not so much a counter signature for robustness, at least among the properties I investigated. And we also showed an example where a structure-based approach led to a stronger predictor than a sequence-based approach. And you really could not have approached this problem without including the molecular features and many diverse molecular features. We saw experimentally and in theory that ribosomal proteins seem to be especially susceptible to this type of damage. And we also saw, when comparing E. coli and radiodurans, that there doesn't seem to be a broad global trend that differentiates the protein structures in terms of vulnerability to oxidation. Rather, there may be some key proteins that have functional roles in recovery that are different between sensitive and resistant species. And I'd just like to acknowledge, I won't go through everybody who was involved in this collaborative work, but I'd like to really thank my advisor, Pam Silver, for giving up her spot speaking to you guys at this mini symposium, giving me the opportunity to do so. And of course, my funding source, the Life Sciences Research Foundation postdoctoral fellowship, which was sponsored by the Gordon and Betty Moore Foundation. And I'd really like to give a special thanks to Ricky Chen for making sure and going out of his way to ensure I was here to speak to you guys today. Thank you very much for your time. Any questions? Yes.
In your structural modeling pipeline, you've probably done this, but I just wanted to check, did you do any blind tests with the ribosomal proteins from Deinococcus that are, where we know the crystal structure?
So I didn't use that to validate the pipeline. I simply used the existing crystal structures in cases where they did exist. I guess indirectly I included them when I showed the sort of global trend of satisfying the criteria that I used for best model selection. As you saw in satisfying those criteria, the actual PDB structures for the most part resemble the performance of models. Again, that's in a broad sense, not on a protein by protein basis. Other questions?
So other types of questions, you have a particular class, you said your favorite class is radioresistance.
Yes.
So how global is this approach, right? Can you...
That's a very good question. It would take a while to investigate that, but I suspect, so this phenomenon probably does not depend on homology very much at all, which I've shown somewhat empirically here. I would think that if you applied a similar approach to something that does directly tie to evolutionary constraints, like protein phosphorylation events, for instance, and how that distributes across a proteome, I actually think that might be a more tractable problem. This is a deleterious and non-enzymatic modification. So I think it's actually a bigger challenge to make this kind of prediction based on structural properties than it would be to address something that has a native function in the cell.
Would you know whether that has been done before, or is this kind of a...
So there have been some attempts to predict phosphorylation specifically, although it has been less structural and more sequence-based again. And I think part of the problem there is people's inherent mistrust of structure modeling approaches, which is definitely founded, but at the same time really limits the types of features that you can incorporate into a predictor to sequence motifs, largely.
Totally naive question. One thing I think you said is that you couldn't take a radiation vulnerable bacterium and engineer it so that it was resistant. But how about the opposite? I mean, it was the second, if you go to the previous slide, I'm sort of keying off of the robustness line.
What I mean to say there, I do think there is a potential to engineer a robust species. What I mean to say is that on the molecular feature level, there doesn't seem to be a signature for robustness itself in the way there does for vulnerability. I do think that there is the potential to try to engineer, either by gene replacement or on the single protein level, to simply get rid of the most vulnerable sites. But the point seems to be that you'll never completely rid a proteome of residues that could, under some condition, get oxidized. You can simply reduce the propensity.
But how about the opposite direction? How about taking your really resistant bug?
Oh, and going the other direction. I do think there's a potential for that, and I think that would be a great validation for some of the predictions I'm making, and I have plans to pursue that in the future.
Very cool.
Any other questions?
Thanks, the speaker again.
Thanks.